This next section of the Unit 4 lecture, we're going to cover a group of organisms known as the non-fermenting gram-negative rods. So just kind of generally speaking, largely the aerobic gram-negative rods can be categorized into two main groups. So we've got the fermenters, which ferment carbohydrates, and those are the ones that we talked about in Unit 3, that family of organisms known as the Enterobacteriaceae. Most of those are lactose fermenters. There were a few exceptions, if you remember. And then the other group is the non-fermenters, which do not ferment carbohydrates. They're going to be lactose negative. So those are the group that we're going to talk about in this next lecture. So some of the general characteristics of this group: they are non-spore formers. They prefer an aerobic environment, and some of them will not grow anaerobically at all. They're non-fermenters, non-oxidizers. So their TSI reactions are usually going to be K over K because they don't ferment those carbohydrates that are in the media. So you're going to see alkaline reactions in the butt of the TSI and the KIA, and they're referred to as asaccharolytic. They will also fail to acidify your OF medium that's overlaid with oil. Largely speaking, they prefer a moist environment. So you can find many of these organisms in natural water sources, in soil, and decaying vegetation. So in hospital settings, they can be isolated from respiratory care equipment like nebulizers, water baths, sinks, catheters. They can also be found in some food. Most of the time, these organisms are going to be considered opportunistic pathogens. So rarely, if ever, these organisms are a part of the normal flora, but they can easily colonize hospital patients, especially those who are immune compromised. Most of these organisms are going to be oxidase positive. And remember, oxidase was a test that we learned about, I think, in Unit 2. So most of them will produce a positive reaction for the oxidase test. There are a couple of exceptions, but for the most part, this test, the oxidase test, is what differentiates this group of organisms from the Enterobacteriaceae. Most of these organisms show resistance, so many of them can withstand certain treatments. They tend to be resistant to multiple classes of antibiotics and to disinfectants. Most of them grow on MacConkey as lactose-negative colonies, and EMB as well. And then the morphology on your gram stain, you'll see thin gram-negative rods or coccobacilli on the gram stain, as opposed to the Enterobacteriaceae, where the gram stain of those, the gram-negative rods, were kind of little fat gram-negative rods. Okay, some clinical infections that these organisms are mostly associated with: septicemia, meningitis, osteomyelitis, wound infections, and wound infections especially following surgery or some type of trauma. They account for about 15% of all gram-negative rods isolated from clinical specimens. You're going to see them usually in hospitalized patients or those who have recently been discharged. And some of the risk factors for infections with these organisms: some type of immunosuppression. So if the patient is diabetic, if they have cancer, if they've undergone an organ transplant, even steroid use. Some type of trauma. So examples of that would be a gunshot or knife wounds, surgery, burns, foreign body implantation. So that could be catheter, prosthetic devices, corneal implants, or contact lenses. And then they're also associated with infused fluids. So that could be saline irrigations or dialysis. Okay, so getting into some of the actual organisms now, the first one we're going to talk about is Pseudomonas aeruginosa. And the Pseudomonas species can be divided into fluorescent group and non-fluorescent groups. So Pseudomonas aeruginosa is in the fluorescent group. So members of the fluorescent group produce a pigment called pyoverdin, which fluoresces under shortwave UV light. Pathogenicity: associated with this organism. This one is the most frequently recovered non-fermenter. It is an opportunistic pathogen, so it's the leading cause of nosocomial respiratory tract infection, not common as normal flora, and it usually is found where moisture accumulates. So it can be in the ears, eyes, catheter, around the catheter, associated with burns, wounds. It can also be seen in nosocomial UTIs, endocarditis, bacteremia, something called hot tub syndrome, and then it's also associated with swimmer's ear. It's associated with pneumonia, specifically in cystic fibrosis patients, and specifically mucoid strains of the organism. It's resistant to most of the antibiotics used for your other gram-negative bacteria. Virulence factors, so it possesses Fimbriae, which you guys know by now helps with adherence. It can have a capsule, form a capsule, which makes the colonies mucoid, and then pili, which also help with adherence. It produces endotoxins and several exotoxins, proteases, homolysins, lecithinase, which is an enzyme that breaks down lecithin found in nervous tissue, elastase, which is it's another enzyme, but it helps it destroy elastic tissue, DNase, exotoxin A. So this one produces a lot of different virulence factors. Lab identification. So the colony morphology on this one, you usually are going to see large, flat, sometimes mucoid colonies. Most of the colonies are going to be beta hemolytic on your blood agar plate. They can have a blue-green metallic sheen on blood agar that I think is pretty. It kind of reminds me of fish scales. And it has a sweet or grape-like odor. It's oxidase positive and catalase positive. It is motile monotrichous. And this one can produce some different pigments. So pyoverdin is a yellow-green or kind of a yellow-brown color. Pyocyanin is blue. No other gram-negative rod produces that pigment. So if it's found to produce pyocyanin, then it would be definitive for Pseudomonas aeruginosa. Pyorubin, which kind of is a reddish brown. You don't see that one very often. Pyomelanin, which is a brown or black. Again, you don't see that one as often. When pyocyanin combines with pyoverdin, that green color that's characteristic of Pseudomonas aeruginosa colonies is produced. And this one also has the ability to grow at 42 degrees Celsius. So there's a picture of Pseudomonas aeruginosa, and you can see one single isolated colony there by itself. How large that colony is, it's very flat. And then up here where the colonies are kind of overlapping, you can see that kind of silverish metallic color that it produces. Okay, Pseudomonas fluorescens and putida, these also are in the fluorescent group. These rarely cause disease. They're associated with catheter-related sepsis in cancer patients. These can grow at 4 degrees, but will not grow at 42 degrees. So that can be one way to differentiate it from aeruginosa. It's linked to transfusion-associated septicemia. And this one produces the pigment pyoverdin, but not pyocyanin. Pseudomonas stutzeri, which is in the non-fluorescent group. This one is a rare isolate. It's an even rarer pathogen in the clinical lab. Most of the infections are in immunosuppressed individuals. The colonies on this one are kind of wrinkled and leathery looking. It produces a light yellow to brown pigment. So there's a picture of the fluorescence that those species in the fluorescent group can produce. And there's an example of those wrinkled leathery colonies that stutzeri produces. So a very, very different colony morphology from Pseudomonas aeruginosa. Okay, the next organism is Acinetobacter. This is the most commonly recovered non-fermenter after Pseudomonas. The two most commonly isolated strains are Acinetobacter baumannii and Elafii oxidase negative. So this is one of those that is an exception to that oxidase rule. It's non-modal, catalase positive. It will grow on McConkey. This one's associated with nosocomial infections. It's considered an opportunistic pathogen. Hospitalized patients can become easily colonized with this organism, but it's also normal skin flora in about 7% of the population. With the increase in antimicrobial resistance seen in this organism, the clinical significance when it's isolated from the respiratory tract or urine specimens in hospitalized patients should not be dismissed. Some species or strains can produce a purple hue that can cause them to be confused with lactose fermenters if you're looking at the colony morphology on a McConkey or EMB plate. Okay, the next one is Stenotrophomonas multifila. This organism was formerly a member of Pseudomonas, then it became a member of Xanthomonas, which is plant pathogens, and now it's Stenotrophomonas. It's the third most commonly isolated non-fermenting gram-negative rod. It grows at 42 degrees Celsius and it can produce a yellow pigment. It's found in the environment, so especially water, sewage, and plant materials. It's very common in hospital environments as a contaminant. It's not normal flora. It can quickly colonize the respiratory tract of hospitalized patients. It is oxidase negative also. It's non-fermentative, catalase positive. This one can appear kind of bluish on McConkey. And this one's implicated in infections of wounds, the respiratory tract, urinary tract, and blood. Burkholderia cepacia complex. So this complex consists of 18 distinct genomic species, all of which have been isolated from human specimens. It causes severe pneumonia in cystic fibrosis patients and patients with chronic granulomatous disease, which are tumors full of white cells. Some of the species in the complex are plant pathogens that have become more opportunistic pathogens in humans. So we're seeing that those recovered in increasing frequency. It has a weak, slow, positive oxidase reaction. Most strains will oxidize glucose. Most strains do not reduce nitrate to nitrite. And then it is modal. Burkholderia mallei, so it causes a disease called glanders, which is a respiratory tract infection, primarily infecting livestock. It's rare in humans. And infections just in general are rare in humans with this organism, but it can cause some acute pulmonary infections, and this one is considered to be a potential bioterror agent. Next is Burkholderia pseudomallei. This one causes a disease called melioidosis, which is an aggressive granulomatous pulmonary disease caused by ingestion, inhalation, or inoculation of the organism with metastatic abscess formation in the lungs. That disease can lead to overwhelming septicemia. It's found in water and muddy soils in Southeast Asia, Northern Australia, and Mexico. This one produces non-fermentative wrinkled colonies. You'll see bipolar staining on the gram stain. So again, that looks like safety pins on the gram stain. It's slow oxidase positive. This one is also considered a potential agent of bioterror. Isolates from someone who has not traveled to an endemic area are reportable to local health departments. And there's some of those colony morphology of Pseudomallei. And next is Moraxella cataralis. This one, the gram stain morphology, you'll see tiny gram-negative coccobacillus and bacilli. It's non-modal. It's oxidase positive. This one is strictly aerobic, which means it will not grow in an anaerobic environment at all. This organism can be confused with Neisseria, and if you remember when we talked about it, I think it was in Unit 2 when we talked about respiratory pathogens, The way that you would differentiate it from the Neisseria is that Moraxella does not utilize any carbohydrates. It can be normal flora of the mucous membranes and of the skin. If we're isolating it from clinical specimens as a pathogen, it would be usually in the respiratory tract, so respiratory cultures, and maybe ears. This one's usually susceptible to penicillin, which is unusual. It's an unusual quality for the non-fermenters. There's some of the colonies of Moraxella. Okay, Olagella urethralis. On the gram stain morphology, you'll see small paired gram-negative bacilli or coccoid organisms, usually isolated from the urinary tract where it can be a commensal. This one's non-modal, and it usually will not grow on MacConkey. It's oxidase positive and glucose negative. Alcaligenes faecalis, tiny gram-negative organisms on the gram stain. It's paratrichous. It has paratrichous flagella. It will grow on MacConkey, and it's oxidase positive. Faecalis is the species usually recovered in clinical isolates. It can be found in soil and water sources, and it's kind of inherently resistant to disinfectants. It's considered asaccharitic, and if you remember, that just means that it does not utilize sugars. And they say that it has a strong apple cider odor, which I've never noticed. But okay, next in the Flavobacteriaceae family, so these organisms are actually weak fermenters, but their reactions are delayed, so they're initially going to appear like a non-fermenter on your media. So in this group, we've got Chrysciobacterium, Sphingobacterium, Elizabeth Kingia, Myroides, and then there's a few others. Members of this family are ubiquitous in soil and water. They're not considered normal flora. Isolates a lot of times will contaminate hospital equipment and can be important causes of nosocomial infections. All of those are oxidase positive. Elizabeth Kingia meningoseptica is the cause of most diseases associated with this family of the Flavobacteriaceae. It can be associated with meningitis or septicemia in newborns, especially in conjunction with prematurity. In adults, Elizabeth Kingia can cause pneumonia, endocarditis, bacteremia, and meningitis. And then the last organism in this group, Shewanella. So this one has been recovered in stagnant water, natural gas, brine, which is salt water, and spoiled dairy products. Its colony morphology, a lot of times it can be very mucoid. It can cause a greenish discoloration on the sheep's blood. This is the causative agent of black fish rot or fin rot. So if you've ever kept fish or been to a pet store and you're looking at the fish and the edges of their fins look kind of jagged or torn, that might be due to Shewanella. They're modal, ONPG positive, nitrate positive, oxidase positive. This organism produces profuse amounts of hydrogen sulfide. And then the species Shewanella algae is halophilic, which I don't know if you remember from, I think it was Unit 1, means that the organism is salt loving, so they can survive in increased environments with increased salt content. This one's not commonly isolated from clinical specimens. When it is, it's usually in a mixed culture, so meaning that there are other organisms recovered at the same time, and it's rarely considered pathogenic. So that's the last little bit, or that's the end of this lecture on your non-fermenting gram-negative rods. The next lecture will be longer, and it's on a larger group of organisms, just kind of some miscellaneous gram-negative rods.